Good afternoon, student. In today's lecture, we are going to start MBT 201 Genetic Engineering subject. So this subject has 4 credits. So I will teach you module number or unit number 3rd and 4th. Rest of the module is shared by Rajashri ma'am. So the 3rd module is different types of PCR techniques. So from today's lecture we are going to start this 3rd module. So in that we are going to see what is PCR, what is the principle, which types of enzyme are used. Then in types of PCR we are going to deal with all the types like real-time PCR, reverse transcription PCR, hot start PCR, then asymmetric cloning of asymmetric PCR, cloning of PCR products. Then after that we are going to see sequencing methods. So now you know the PCR technique is very important as this is used for coronavirus infection. That is the type of PCR used is RT-PCR. I think you are familiar with that. So after completion of this module we will see the 4th module, that is gene manipulation and protein DNA interaction. So in that we are going to see mainly the libraries, genomic libraries and cDNA synthesis libraries. Then cDNA synthesis is very important. And then other technique. So for this subject you can use these books. So I have uploaded 4th ebooks on your Google Classroom. So you can use that. I will suggest you to use a molecular cloning or laboratory manual by Sambrook and Russell. So this book is very important. So that will help you in understanding the practicals. So now we are going to start the lecture. So first we will see what is PCR. So PCR is polymerase chain reaction. So why it is called as polymerase? Because there is use of polymerase, DNA polymerase enzyme. Then why it is chain? Because in this reaction what happens? The substrate, the product which is formed in first cycle, so that will be used as a substrate in second cycle. Keep in mind, or in second reaction. The product which is formed in first cycle, that is utilized as a substrate in second cycle. So that's why it's a chain reaction. Keep in mind, right? So what is PCR? PCR is a technique where the DNA is used, or the specific sequences of DNA is amplified. So what is amplification? That is production of n number copies of DNA. That is amplification, right? So what is PCR? It's a technique where specific sequences of DNA is amplified and that is further tested. So the PCR products also analyzed using gel electrophoresis. And now so after finishing of this whole topic we are going to see the application. Now you know the one of the very important application is disease diagnosis. And in this PCR we are going to use obviously DNA molecules and the enzymes. Enzyme is polymerase enzyme. And this technique is first invented by the Nobel laureates Mullis in 1983. So this Nobel Prize winner is from chemistry. So now we will see the components of PCR reaction. So first of all, what is needed? Target DNA. So target DNA whose sequence is amplified, so that should be present, right? So that is target DNA. Then pair of primer, right? So this is short oligonucleotides, right? So that is used for DNA amplification. So there are pair of primer, forward and reverse primer. Keep in mind forward and reverse primer. You should know the direction of forward and reverse primer. So this is the question for you, the direction of forward and reverse primer. So you have to comment in you have to give answer in comment box. So your class does not answer any question asked during the lecture. You should answer this question in the comment box, right? Then DNTPs, that is deoxynucleotide triphosphates, right? That is the nucleotides. So if you have to amplify the DNA sequence, target DNA sequence, right? That means you are going to make n number of copies of target DNA. So what is required? There should be nucleotides, right? So there are DNTPs. So next is thermostable DNA polymerase. Keep in mind the DNA polymerase which is used in PCR, that is thermostable. Means at high temperature it will not denature. So its activities remain same. So the activity of DNA polymerase will not affected at high temperature. So that's why the thermostable DNA polymerase is used in PCR. Then Mg2+ ions, right? Magnesium ions. So as we are going to use DNA polymerase for this reaction. So I think you have studied biochemistry. So for activity of the enzymes there is requirement of cofactor. So for DNA polymerase there is magnesium ion that will act as a cofactor. That is very important for DNA polymerase activity. Then buffer solution, right? So what is the role? So maintain the pH and ionic strength of the reaction. So that is suitable for the enzyme activity. So this is about the components of PCR. So that is about the chemicals. Then other than that, what is in case of instrument? So this is the machine is used for PCR. So that is called as thermocycler. And now so these are the enzymes. These are the some components which is required for PCR reaction, that are all added in this PCR tube. And then this PCR tube is placed here in this thermocycler. Then program is set. So we are going to see the program of PCR. Then the cycles is going to on. So this is thermocycler instrument. So now we are going to see the PCR principle or the process of PCR. So this process comes with 3 steps, sequential 3 steps. Keep in mind, that is denaturation, annealing, extinction, and repetition of this cycle. Denaturation, annealing, and extinction. Keep in mind, this sequence is very important. So now we are going to learn what is denaturation, right? So what is denaturation? Actually there is breaking down of the hydrogen bonds which are present between 2 DNA molecules, right? So there are 2 DNA molecules, 5' to 3' and 3' to 5', right? So this is I think you know the structure of DNA, right? So there is in denaturation what happens? There is breaking of the hydrogen bonds which are present between 2 DNA strands, right? And after what happened, after breaking the hydrogen bonds, the DNA molecule will become separate. They will become single stranded, right? The DNA is double stranded, right? So there is presence of hydrogen bond, right? So how this takes place? Because of higher temperature. Keep in mind. So you can see the temperature here 94 to 98 degrees Celsius, and this temperature is for, keep in mind, 20 to 30 seconds. So this is going to vary the temperature and time as for during the practical. Keep in mind. So this is not the or you can say standard. So the program is standardized during the practicals. Keep in mind, right? So this is theoretical we are learning. So there is denaturation of DNA molecules at high temperature, right? So generally this temperatures is for 94 to 98 degrees Celsius for 20 to 30 seconds. So this is the first step of PCR cycle or PCR cycle. Then next it's annealing. So now in this step what happens? The temperature is lower up to 50 to 65 degrees Celsius for 20 to 40 seconds. So that will allow the annealing of the primers to the single stranded DNA molecules. So you can see in this, so this is first diagram. You can see this is a double helix of DNA. So at the in denaturation process, what happens? There is breaking down of this hydrogen bond, and you can see separation of DNA strands in a single stranded. Then next step is annealing, where there is binding of the primer to the DNA. You can see here. So these are the reverse and forward primers. You can see there is binding between these. And these are short oligonucleotide nucleotide chain. You can see. So there is like a DNA, it's not longer. So you can see it's short nucleotide DNA fragment, right? So there is annealing between these two, between primers and DNA. So binding means what happens? There will be DNA hydrogen bond formation between the primer and DNA sequence. So this is about the annealing. You should know the temperature. So next is extension, right? So in this process, the DNA polymerase comes into the picture. So now you know what DNA polymerase will do. So most of the time there is uses of Taq DNA polymerase. So this is the DNA polymerase which is used commonly, and its optimum activity at 75 to 80 degrees Celsius. So what happens in this extension process? So Taq DNA polymerase will extend the DNA length by adding DNTPs. I think you know all the process like replication. What is in by replication? Transcription. What is the role of factors, polymerase, helicase, ligase. So as I told you, the primer is extended by adding the nucleotides complementary to the DNA, right? So extension, what happens? There is completion of first step, right? Then the second cycle will continue. So generally there are 30 to 40 cycles. So you can see, so this is the length of primer. You can see in the extension process, the length of this primer is extended by adding this DNTPs, and that addition is done by DNA polymerase. So in exam they may ask you the after completion of 15 cycles or 30 cycles of PCR, how many copies will form? So there is formula 2:n, right? So n is the number of cycle, right? So after you can see at 2:2, right? So initially there are 2 strands of DNA, right? And every cycle the copies is going to double. So that's why it's 2. Sorry, that's why there is n. Then you can add the number of cycle at the place of n, and you will come to know the copies of DNA or PCR products. This is all about today's lecture. In next lecture we are going to discuss further. Thank you.